Gonna mark your work now. Gotta do some work. All right, it's a great pleasure to have Professor Hilgemann, Don Hilgemann, who is originally from Iowa, but it's true. But he's joining us from Dallas, Texas, where he's a professor of physiology in UT Southwestern in Dallas. So welcome to St. Louis. I first encountered Don in his work when I was looking for experiments and formulations of the sodium-potassium pump and sodium-calcium exchanger in the context of our own development of whole cells in cardiac myocytes. And that's where I saw this, his beautiful and creative work and very highly quantitative work. And since then, we've known each other and exchanged ideas over the years. Dan is very original in his work. He's doing beautiful experiments on transporters in cardiac cells. In particular, the work that I know about the sodium-potassium pump, sodium-calcium exchanger, and I think sodium-hydrogen also. And more recently, he's looked into regulation of these processes through endoexocytosis and also PEEP2, which is popular on this floor. So, Dan, thank you for coming.
Thank you. Great. Thanks, Emil, for the very kind invitation and for the cordiality, and have a wonderful day and lots of wonderful food and wonderful weather, a nice place. Thanks for your help. I'm just going to jump in. It's like being, I have friends here. It's like being home, so I'll just jump into what I want to talk about, which is coming back to one of the themes which interested me early on. It gets rather little attention in the bioelectricity field, and this is cardiac. But the bottom line is sodium-potassium pumps are the core machine that set up ion gradients that allow ion transport to happen. And I thought that before I would retire, I decided these are so important. I'm going to go back to the old theme which interested me from early on and try to figure out something about their regulation. And you will see as I go along why that is. This is a substitute really for studying their function, and believe it or not, we still have a lot of real holes in our knowledge about how these pumps function. So here's the way they would look in a cardiac myocyte based on recent crystal structures. This is inside here. Here's the famous aspartate that gets phosphorylated at each cycle. There's a domain which is a phosphatase, and there's a domain which acts like a kinase. And so they are self-phosphorylating, dephosphorylating enzymes that here use the cytoplasmic transmembrane domain to move on average 2 potassium in and 3 sodium out at each cycle, therefore electrogenic, making a continuous outward current in the cardiac myocyte. The reversal potential is probably about minus 180 in a myocyte, so they're never going to reverse their function really. They're just working ahead, but they get a bad name for just being a workhorse because they're much more interesting. Okay, they have a beta subunit, much more relevant to us. They have a quasi-gamma subunit called phospholemma in muscle, FXYD proteins, which I believe interested Dr. Nichols at one point for their putative ion channel activity. Anyway, and this thing gets phosphorylated several times, therefore is thought to be the regulator of this channel, and also has 2 formidolation sites on it. So I have been interested in that, but it's going to be very little of that in my talk because the regulation, which to me now appears highly physiological, actually does not seem to involve any of it. So here's the cycle. And this is why I say this is a substitute for what I really want to study. This is the way the pump works, quasi the transmembrane domains. Outside is up, inside is down during each cycle. So let's start down here. When it's open to the cytoplasmic side, it can bind 3 sodium ions. We were all so happy to see that the 3 ions bind in parallel. You know, a lot of people like to say pumps and transporters. No, channels and transporters, they're all the same, you know. And you just modify them slightly. It's really not true. And I don't go with that. But it's very impressive. The ions bind in parallel, not really in series. And then it phosphorylates itself at the expense of an ATP, closes on the 3 sodium with the phosphate covalently bound to that aspartate, opens to the outside. One gets off in a highly selective low affinity site, then the others get off. Those sites bind to potassium, and in the absence of this other site being occupied, it closes on the potassium, dephosphorylates, and comes back to the cytoplasmic side to bind ATP and make another run. It's an amazing beast, and we're very far from having something like, molecular dynamic simulations of the whole cycle, because there's many reactions that depend on other things, or that depend highly selectively on, certain events having occurred. Now, this sucker is interesting. So in a typical cell, of course, it's making the ion gradients you use for so that potassium can continuously leave the cell and hyperpolarize it. Sodium can come in to make an upstroke, and so forth. And it also makes the energetic investment needed to make a chloride gradient by either driving chloride out through chloride channels or by letting it go out with potassium in neurons. So it's just a workhorse, but if you stand back and think about it, boy, it has to be somehow highly regulated. So here's what we're talking about. In muscle, it's not so impressive. If you calculate how much of the energy of a skeletal muscle is being used by the pump, it's 5 to 10% at rest, and then during activity you go up. Not that impressive, but in the brain, for sure, it's more than 50%. In the kidney, it's almost everything. Almost all the energy being used by the kidney is being used to pump ions. And in the liver, 20 to 25%. And that's impressive because we're talking about 30, or even sometimes people say 40% of total resting ATP utilization is by this pump. And actually, this liver part is interesting because although that's only about 6% of tissue, actually, the liver and the gastrointestinal tract is accounting for at least 30% of basal energy use in our body. And this for pumping ions. That means that for sure, how the pump does this is important for the final calculation of thermogenesis. It's been a question for 50 years whether the pump is involved in thermogenesis, and that question is still not answered. And this would all presumably affect how the body might go about regulating the pump. So, energetic efficiency for sure can be regulated in mitochondria, if nothing else, by making cycles of protons, to make heat. So for sure it can be regulated in mitochondria. It's still a question if F-ATPases are really can be changed to be, thermogenic or not. Calcium ATPases have been implicated to be thermogenic, and different isoforms can be used apparently in thermogenesis to generate more heat or not. So, sodium pump, all of this basic question is still not very clear. what was clear from the olden days when people did calorimetric measurements of energy use, they could accurately figure out how much energy is the pump really using. And we electrophysiologists have become very primitive. And I think biochemists have become more primitive. They just say the pump is a 3-to-1 ion pump and uses 1 ATP per cycle. But this was well within the resolution of these kind of measurements. And actually what you come up with is more interesting than that. Stoichiometry is not perfect. Biochemists always claim that. And here we have the pump at very most, with very accurate calorimetric measurements, can pump about 2 sodium ions per cycle, not 3. So for sure things are not as simple as we were saying. So I would like to work that all out. And somewhere in the regulation of the pump, that must go into things. So for calcium pumps, it's quite clear that every now and then you have failed cycles and you hydrolyze ATP but don't actually pump ions. Things like this are presumably going on. It's always been of interest whether, like, every now and then this 2-sodium bound state can be dephosphorylated and fall back here so that you'd be pumping 1 sodium per cycle. There's plenty of possibilities for complexity. But you would imagine evolution would say, let's think about how this gets regulated. But now I'm going to get more primitive. We just want to know if the activity of this pump is regulated. It's claimed that it's cyclic AMP kinase mostly, a little bit PKC in the heart. That's about all that's been really worked out for the heart, where the pump continuously makes these gradients that you need, not only to get potassium out, to make repolarization, and to get potassium in so that you can let potassium out, and to get sodium out so that you can let sodium in to make the upstroke, and also to extrude calcium. At the beginning of the cardiac cycle, some sodium's coming out, some calcium in by sodium-calcium exchange. And later in the cycle, calcium's coming out and free sodium. Now, as Yor mentioned, I worked on sodium-calcium exchangers a lot, but I came around that the pump is more important for me to use the rest of my career on. Why? Because when you look at how these things work together, this is the situation in the heart which interests us the most. Sodium-calcium exchangers are always trying to move calcium toward some equilibrium that is determined by sodium gradient and whatever electrical potential is doing. And it worked out in many different knockouts and in the development of drugs which can block exchangers. What you find is, as important as this is, you change the number of sodium-calcium exchangers and you really do very little to EC coupling. So you can knock down sodium-calcium exchangers by 90%, and here this calcium transient almost does not change, very little in magnitude. The heart is fine. You can overexpress 2 or 3-fold, and the same thing. And that's because it's really just striving to get calcium to some equilibrium. But that equilibrium is determined by what? It's determined by sodium flux in and out. And the biggest influence is this pump activity. So the way it works out, the pump is a master and the exchanger is a slave. So that if you change pump activity by a small amount, 5 or 10%, you get a huge effect on contractility, like doubling, halving. And if you do simulations, at least in my hands, I will ask Europe, of cardiac EC coupling, and then you go through, you change the activity of every important protein that affects calcium, the biggest effect you get if you change activity of each of them is the pump. So if you change activity of the pump by 5%, you get a huge effect on contractility, okay? Whereas you can change almost all these other players Cyanidine release channels, calcium pumps here in the SR. They're very important, but not that important. So
When you change those, do they get to the right organelle? When you say there's no difference in function after you, right?
after we overexpress
Or
and
knock
after
down.
we can see that. So I have a mouse. Now, don't get me wrong, the exchange is important. We actually, so my buddy Ken Phillipson made mice where for sure we could get the sodium calcium exchange activity in the myocyte up by 2 to 3-fold. We have made a mouse, we have 10-fold increase. The mouse is fine, but don't get me wrong, for the electrophysiologist, if you do something nasty, if you shake the mouse up a little bit, it can go berserk and we'll get all kind of arrhythmias and can fall over dead. But anyway, what I'm really talking, we're up to 10-fold overexpression. So yes, we can get them. Okay, so this is what pump currents look like when you study them in cardiac myocyte. And I'm showing you here, this is membrane capacitance over time. We're always making square wave voltage pulses from which we can determine capacitance, the actual conductance, and then the actual membrane current. So you take this as mouse myocyte, typical mouse myocyte. There's one thing a little exceptional about the condition here this is my basal condition. I use 25 millimolar sodium inside. There is 90 millimolar potassium inside and 20 millimolar TEA. A lot of TEA outside, some NMDG, very little or no sodium outside. So this is a clean condition. And the reason we use this condition, in this condition, the pump is not fully activated by the sodium inside. There's antagonism between potassium and sodium. So even though it's quite high sodium, for sure this is not maximal pump. The way we turn the current on, we put potassium outside, we get this outward current. And the characteristic thing is that the current turns off. By 50, over time, over 15 seconds, maybe 60, 70%. Then you take away the potassium, and it's really off. And you can repeat this again and again. We're very comfortable. This is a clean situation because here we have to use a lot of Wabane in the mouse, but you can still nicely block the pumps. So you put the Wabane on, and so here we have potassium on, take potassium away, and afterward the current's really gone. So we're very comfortable. This is a clean measurement of sodium pump, although most people don't put potassium inside. Okay. The curious thing we knew about this like 20 years ago was that in the intact myocyte, the current has this property that it decays. And we always thought, everybody would think, okay, you're pumping the sodium out or you're depleting the ATP or something. That's why the current decays. But actually, working on sodium calcium exchangers, we found out that it's not changes of ion, it was really an inactivation mechanism. And so we had in the back of our mind a long time that question, what is going on in these measurements? If you simulate this situation, this is a half nanoampere roughly, you will find that it's questionable. And you simulate, our pipettes are pretty big. Our access resistance is always less than 3 mega ohms during the recording. If you set it up and simulate the situation, it's at least doubtful that sodium is being depleted. But it could be, but let's have a look. Let's do something to find out. So, as an aside, we also can study the pump in large excise patches from cardiac myocytes, actually 20 micron in diameter. And when we do that, then we put potassium outside in the pipette, sodium inside, and we look here and see if there's any transient behavior, kinetic behavior, when we change ions. So we're starting here with no sodium, 0.6 millimolar, 40 millimolar back, up, down, different. And what you just see is, in this situation, it's just square. There's no kinetics in the excise patch, so that's why we didn't pursue this earlier. There is one kinetic only. That is, the first time, when you pull this giant patch off of a myocyte, there is one place where there's a kinetic, and that is the very first time you apply ATP. So here there's no ATP, but there's sodium, and now we put ATP the first time. And here what you see is you get an immediate current, and then it turns on slowly over a couple of minutes. That's the only thing we know about kinetics in the excised patch, which makes us suspicious. We worked on this a long time, but this must be very important, some kind of phosphorylation or lipid change or something. And no matter what we did, basically, after you do this once, you can turn it off. You can wait 15 minutes. Afterward, it's a square wave, just like I showed. So this is one indication in the intact myocyte. We were thinking there must be some kind of inhibitor which can float away. And that might be why the currents are transient in the intact myocyte. There's something which can bind and float away. Okay. Sorry.
Doc,
Yeah, just go ahead and ask.
yeah, so did you see the same thing if you, before you size the patch, you activate it
Yeah. If you have ATP in the cell,
in the cell?
it's still, I mean, well, it's fairly quick. So you have to yank it off and get it in the chamber, and you see it growing. You definitely can see it growing always the first time. So even if you take a healthy myocyte, you get your patch, then you're in the chamber and you shoot solution at the myocyte, you knock it off, you see the pump current turning on. That's why we were thinking a long, long time there's an inhibitor in the cell which floats away. We never identified it. Anyway, now we're going back to it. Okay, back to intact. Is this ion depletion, ATP depletion, or something more interesting? Oh, come on.
Just tell us what it is.
All right. So the easiest thing to check about ion depletion is we just do this at 37 degrees, and we go to room temperature, or even a little bit below. You decrease the amplitude of the current by 5 or even 6-fold, or let's say 5-fold. And what you see is you still have these current transients. What that tells you is, so the current decay, the fractional decay is just the same when activity is decreased by 5-fold. And that's already pretty good, I think, evidence that it's not sodium depletion. Is it ATP depletion? So you can calculate how much ATP you need. Well, first of all, the pump works with a few micromolar ATP. So, you know, in general, ATP is not going to fall to a few micromolar when we use 8 millimolar in the pipette. But let's do something to test that. So what we do, we look at this behavior at all different sodium concentrations. We start with a saturating sodium concentration, then we do it with 20, 10, whatever it is, 10, 5, and 2.5 millimolars. And this is at room temperature, so the currents are quite small. And now we look at the fractional decay. So what you see is if you go to high sodium, and if you get up here to even 80 millimolar sodium, there's almost no decay. Peak steady state is almost the same. And the decay gets larger and larger as you decrease sodium. So this is what it looks like. This is the fractional decay. And you can fit a curve to this data that really goes right up to 100% decay if there's no sodium projected. And that looks the same under our other conditions. Basically, you can fit this data to very simple models. It looks the same when here we're at 37 degrees with no potassium inside, steady state and peak. And here's with potassium inside. Well, now it's not so obvious this is going to get up to the other peak, but you can fit it with equations that actually would do that. And what kind of model would you need to do that? Well, for sure it's not ATP depletion, because ATP would, of course, deplete best when the current is very big. And this is the opposite. The current decays most when the sodium is smallest. Okay, now if you really want to question me, is this ATP depletion, sodium depletion? It turned out I just had a stupid idea. Well, these are conformational changes of the pump. Somehow it can inactivate. Maybe if I put something chaotropic in the cell, I just thought I could, maybe chaotropic things would tend to prevent this reaction. So anyway, that stupid idea seems to work. If I just use like 150 millimolar glycine, that's somewhat a chaotropic solute, then there's no inactivation. And you can see here at high sodium, the currents are enormous, more than 5 picoampere picofarad. And there's no difference, almost no difference between peak and the 10-second current. So,
What
it's
does
a...
chaotropic mean?
Hmm?
What does chaotropic mean?
Chaotropic means you prevent interactions of proteins. Chaotropic means you prevent protein aggregation. You prevent interactions.
Okay.
Chaotropic versus cosmotropic. You're a biochemist, aren't you?
Don't know that word.
Cosmotrope and chaotrope.
How does glycine do that?
Hmm?
How does glycine do that?
Well, people look at this stuff a lot, so you have to go to the Wikipedia and look up chaotrope. So, chaotrope, urea is chaotrope, right? It turns out sugar is not, I think, a little bit the opposite. So
Causing chaos.
causing chaos. So of course it has plus and minus charges on it, and it's got that hydroxyl on it. So somehow this combination is like urea is chaotropic. Urea is chaotropic. Water is not. Very okay. So all right. Let it be said, we know how to get rid of this current decay, thereby eliminating the idea that it's due to depletion of ions or ATP. Okay, how could it be taking place? We said the current decay becomes bigger and bigger when you have less and less ions bound to the cytoplasmic side, and since potassium may promote the inactivation a little bit, but it's not a very big effect. So overall, what we would tend to conclude is that when a site that cannot bind potassium is empty, and that's what we call site number 3, that's the sodium selective site. Whenever that site is empty, you tend to fall into an inactive state of the transporter, and it takes some time, like several seconds, to recover. That would be an exact parallel to what we found for sodium-calcium exchangers, except that for sodium-calcium exchangers, inactivation takes place from a fully loaded state. But I like it because it reiterates a theme of our lab that in transporters you have regulation taking place, independence on ions bound in different states of the transporter. So then let's go on. So that's the way I can write some simple equations to describe what we're seeing. Now, how would you like to test that? Well, here's the simple way. With sodium-calcium exchangers, we could kind of test what we were seeing, because what we could do is with sodium-calcium exchange, we can put 3 sodiums outside. That would be 3 sodium over here. This would be 1 calcium going in. This would be 3 sodium over here. When we put 3 sodium, or when we put high sodium on the outside, sodium-calcium exchangers, you can push to go in reverse, so that they would open to the inside. And we could prove that when you put 3 sodium outside, you could push them backward through their cycle so that they could then bind sodium inside and inactivate from that state. Here with sodium pump, we want to push. Here we want to push all the way this way and see if we can make them inactivate by putting sodium here. But we have the irreversible step in between, unfortunately, which is that ATP gets hydrolyzed here, so to go backward you need to synthesize ATP, and that will not be possible unless you have plenty of ADP. So would that work in the normal state of the cell? That's not at all obvious, of course. So here we're doing pump current. Now, Instead of just taking away the potassium, what we're going to do, we're going to apply the sodium to the outside and see if afterward there's more pumps inactive. So here we have now, we have no potassium and we have sodium outside. So there's inward current, there's some sodium inward current, probably leak current. Now we put back the potassium and take back away the sodium. And what you see is the pump current is even bigger than it was before. Well, maybe some sodium loading or something, but for sure it's not smaller. So for sure, this experiment that worked with sodium-calcium exchanger does not work with the pump. Well, maybe we can do something to make it work? Okay, what would we do? What you want to do is deplete ATP a bit. You need to have ADP in the cell, so we can put a bunch of ADP in our cytoplasmic solution. But the ATP turnover is very, very powerful. That really works quite effectively. If we put 10 millimolar deoxyglucose in our pipette solution, what will happen? Deoxyglucose gets phosphorylated like crazy. There's huge hexokinase activity. We're going to deplete ATP substantially, make a lot of ADP. So now the pump current's running down, because ATP eventually is going down, ADP is going up to be equal to ATP, probably. So our peak current's getting smaller and smaller. Now we do our famous experiment here. We make pump current with potassium. Now we take away potassium, we put sodium. Now here there's a little inward current, of course. Now we put back the potassium and take away the sodium. And you see the peak current is drastically decreased. So in this situation, we're almost for sure there's ADP in the cytoplasm. The pump can go in reverse through those steps, open to the cytoplasmic side, and now you can get your inactivation. So that's the idea. That's our argument. This is real inactivation. So sodium-calcium exchangers use this to regulate. So basically, different things remove inactivation and promote exchanger activity. So of course we were interested whether that's the case in the pump. So here's this usual experiment, and we check the most obvious thing first. That's pump activity, pump activity, pump activity. As I said, the most well-known finding by some people about sodium pump regulation in cardiac myocyte is that it's activated by CAMP-dependent protein kinase, by isoproterenol, anything that activates CAMP kinase. That's extremely well known, and this involves that phospholemma I told you about getting phosphorylated on multiple sites. And the trouble with this thing is that 3 or 4 of us in the field who've looked at this over 20-some years have never seen that effect. So this is my experiment with isoproterenol. There's just no effect. Now the funny thing that really got me about this is that this is the perfect condition where isoproterenol is claimed to activate the pump. The claim is isoproterenol increases the affinity for sodium. So if I need to mention it, this is Dunbar's, and this involves phospholemma. That's Amy Tucker made that knockout mouse 20 years ago. And phospholemma is the most phosphorylated protein in the surface membrane. So 3 or 4 of us have never seen that effect. Certainly I've tried it in excise patches. I didn't see it. And this would be a perfect condition for it. Now why would that be? So all of that work, it's really convincing, it was done as follows. It was not done in voltage clamp myocyte. It was done in myocyte sitting in a dish. And what was done was you incubate the cells with the sodium-sensitive dye. You look at the fluorescence. Now you take away potassium in the dish. The cells load like heck with sodium. Now you put potassium back, and you watch optically the sodium go back down. Then what you do, you plot the rate at which this fluorescent signal is going back to baseline. That's proportional to pump activity. And you plot the sodium concentration. Then you do it with isoproterenol, and what you see is the sodium dependence is shifted to lower sodium. Okay, this is the perfect condition. We see nothing. Now, why would that be? Okay, so first of all, nothing with isoproterenol, peak 10 seconds, peak 10 seconds. Nothing with phospholemma, peak 10 seconds, peak 10 seconds. It's also claimed that PKC activation increases a little bit the peak, increases the peak pump current. There, with OAG as a PKC activator, we see a little something, but it's pathetic. Very small. So, what could have been going on in those experiments? I believe at present it's related to this finding. Here, instead of here's pump current with like 70-some% decay. Instead of trying isoproterenol, we make the myocytes contract a little bit. So what we're doing here, we put calcium outside. Normally we have EGTA in the bath, so there's no calcium whatsoever. We have 2 millimolar, or depending on the experiment, 2 or 4 millimolar magnesium outside. So we just switch that out for calcium. This is now a sodium-calcium exchange current. And then we just turn it on for a few seconds and turn it off. So I should have a movie, but it's kind of boring. What happens is that's enough calcium going in to get the myocyte to start to beat. And then it stops. It stops. What you see now is the capacitance start to increase a little bit. It goes up maybe 5 to 8%. That's pretty small. And afterward, what you see is the sodium pump current, rho in the hole, is increased by a good factor of 2. And the 10-second current is increased by maybe factor 3 or 4. Then this decays back down again. Okay, here's just another feature of that. It started to get very interesting. So what you see is pump current over time will tend to decay. Now we activate exchange current. Here you see in more detail what happens. The exchanger is actually secondarily activated by internal calcium. So what will happen is usually the exchange current is at first very tiny, then calcium starts to accumulate inside it. It turns on more, but slowly. You turn it off. Afterward, pump is highly activated. Peak is steady state. Turn it off. Now, you can wait even 3 minutes or so. The myocyte is completely quiescent. We have a little EGTA in the myocyte, so we're very comfortable the calcium is back down again. And afterward, instead of being this exchange current, you put calcium again and you have this exchange. So whatever is going on activates both pump and sodium calcium exchange. And then afterwards, sometimes the pump is still big. Now, I need to interject that there's still something more that happens with calcium if you repeat. So usually you can repeat this whole sequence 2 times, and sometimes 3 times. But then if you repeat the calcium transient many times, this is what happens. So, all right, so this is pump going down. This is a little calcium, the myocyte contracted a few times. It stops beating. You can tell it stopped beating because the beating makes a little artifact in the current trace. And now, of course, the pump is activated, steady state is increased. The pump goes back down. Now, often if you do another calcium transient, what will happen is then we get this kind of endocytosis we study. So this is a decrease of capacitance. The cell is taking in membrane. It's not particularly big response. It can be even very, very small. But now something else is happening, and afterward here, this is pump current, and then here, this is pump current. The pump can be 100% obliterated by 2 or 3 cycles of what appear to be not very aggressive calcium transients.
John.
So there is a stimulatory effect and there is an inhibitory effect on the pump, and I'm not going to talk about all of this today. Also on other channels we've started to study in the myocyte.
Is the latter effect pinching off of T-tubules?
Nope. we talked about that the last time I was here, this kind of exocytosis is not pinching off the T-tubes. We can be sure to put fluorescent dyes and still see the T-tubes nicely. The vesicles that are forming in this kind of endocytosis are much smaller than a T-tube.
You're missing a little green square underneath that big one in the middle. Click.
Where?
Left, left, left.
Here?
No, left, left, left. There.
There.
Yeah.
Oh gosh, you're right.
Okay.
Sorry. Absolutely.
So,
Good eye. Sorry. You're right. Here. This is pumpkin.
okay, so you're putting calcium on transiently. It's
Yes.
causing big sodium-calcium exchange, which is putting calcium in and taking sodium out.
Mhm.
So it's depleting sodium.
Yes.
So that's why you're getting the sodium pump activation, you say.
Not at all.
So how's it happening?
Yes. Okay. All right. First of all, yeah. So okay, that's my talk is about this effect.
Okay.
Okay. Sorry about that. Here we go. Okay. So first of all, if this is physiological, you'd like it to happen when sodium is low. So here we have a 10 millimolar sodium inside, and actually you want to know that you don't need potassium inside. Here we use cesium instead. That's pump, pump, pump. Now, instead of we cannot use exchangers to get calcium in because that sodium is too low. What we do, we just activate some calcium current. So this is a BK-like drug which keeps calcium channels from closing, from inactivating. So we start with EGTA here. Then we put the calcium. Now this is a calcium current actually. And afterward, pump is activated steady state. So that's not. So we'll skip this. So there's a lot of work saying it's really an effect of calcium. Now, how is calcium doing that? 2 years have passed. 2 years. We started with the obvious. Is it phospholemma? Everything looks identical in phospholemma knockout mice. What was isoproterenol doing anyway to make the pump current increase in those myocyte experiments without voltage current? We think isoproterenol made the myocytes contract a few times. And it was this effect. And it had nothing to do with pKa being phosphorylated and so on. Okay, we're interested in palmitoylation. There's a knockout of the main palmitoylating enzyme. That didn't change this. We're interested in PKCs. Here's Kemptide, organic PKC inhibitors. Here's H7, it's still the same. We're interested in cyclosporin, of course, calcium, no change. We're interested in cytoskeleton, Floyd and Trumpkin, no change. We're interested in ROS. And there are claims the pump is regulated by glutathione.
There's none of that.
Antioxidant cocktail, name, nothing. Peroxyreduction knockout mice, no, nothing. SOD, nothing. DHE. Diacylglycerol-kinases are calcium dependent. Nothing. Bisphorone, very interesting protein in muscle. They get a lot of nothing. Phospholipase D, double knockout phospholipase D, nothing. We really like that a lot. Ruthenium red inside, nothing. Cam kinase, nothing. CK50, that I think blocks mitochondrial sodium calcium exchange. Camptide, nothing. Now you're down to scraping the bottom of the book. Okay? Genistein, complete block at reasonably low concentration. So tyrosine kinase. Then we worked on. Didn't seem to be tyrosine kinase. We tried to do more specifically. Methylene blue, another junk drug that affects supposedly electron transport in mitochondria, completely blocked, and even quercetin. So, what's his name? This biker. This is the same concentration he takes, partly in his blood. What's his name again? Lance.
Lance Armstrong.
Okay, we're down to junk. We're down to hydrophobic junk to block this, after almost 2 years. Is it, then we asked the stupid question, is it the membrane itself that's changing? We actually had good luck with this. That's how we discovered that PIP2 was important. We could prove that the cell was generating negative charges. But in this case, it's nothing to do with negative charges. You can put 10 millimolar polylysine or neomycin in the cell. Everything looks the same. But the membrane is changing. How do we know that? Okay, here we're using a little trick. We decided to look at what we call membrane order. So we look often at how hydrophobic compounds bind to the membrane of cells. And what we know, this is the idea. This is the idea. Membrane order will determine how well a lot of things get through the phospholipid membrane itself. If you have more cholesterol, water can't get through, a lot of things can't get through. Here we use something called a hydrophobic cation, hexyl triphenylphosphonium. It works with tetraphenylphosphonium and many different hydrophobic compounds. So this is the idea. This has a delocalized positive charge. It has 3 phenyl groups. People use this to get things into cells or into the mitochondria even. So it can get through the membrane, and when you put it on the cell, it makes this current here. It's a positive charge, so it's inward current. So what we have shown is it's very dependent on membrane order, what we think of as membrane order. By that I mean if you extract the cholesterol, so the outer membrane becomes more disordered, so it doesn't tend to form domains and be rigid and stuff, this current will go up like 3-fold. And so, okay, so this is just getting through the bilayer. Okay, so this is pump current, pump current, pump current, and in between each one we do this hydrophobic cation. Now here's calcium, that's the outward sodium-calcium exchange current. Here, as always, we have this little increase of capacitance. Here we have the pump current has increased from here, more than double. Steady state current, 3-fold, it's coming down again. And here's the hydrophobic cation current. Not quite double, but anyway, good 50%. And it goes back down exactly in parallel with the current. So we do this many, many times. And you keep going now. Let's say you make another calcium influx episode. What happens next is the pump current is gone. So of course we're very interested in this. You could affect the pump so much. And this hydrophobic cation current also keeps getting smaller. So we've done a lot of work with this, and it's clear that the membrane itself is changing. Now, seeing that the membrane itself is changing, we also ask, is this something more general happening in the cell in response to physiological calcium transients, which might affect other channels? And to keep Dr. Nichols awake, for sure, potassium channels are affected. Now, probably should have shown, oh good, I didn't show it. Now, one thing we knew about sodium pumps, which is kind of strange. People have used fluorescent dyes, hydrophobic dyes, to look at conformational changes of the pump or ion binding by the pump. Now, how would that be possible? So the Germans, starting with Peter Leuger and Hans-Jürgen Appel, for 30 years have been looking at electrostyral dyes in the membrane, how the fluorescence of voltage-dependent dyes in the membrane change depending on how the pump sitting over here away from the dye binds ions. So this is a biophysical problem which is quite interesting. People do not believe that ion channels sense the charge from phospholipids anymore. People believe that the surface charge, what we call surface charge of ion channels, it's really part of the protein. Okay? And you don't see much effect of changing lipids on the conductance of channels. And therefore you don't expect the channels affect, on the basis of the charges, affect the membrane. But for the pump, that's different. And why that is, I don't know. But the fact is, we did the following experiment just to see if we could duplicate kind of the impression of people like Peter Leuger. We asked the question, is it possible that the pump configuration also can affect the membrane like the membrane apparently is affecting it? So here we make this hydrophobic cation current once, twice. Here we make the pump current. We let it inactivate. We think it's in a strange conformation from which it can occur. And sure enough, the next time you make the cation current is down and it recovers again. You can do this again and again. Here's a pump and activation that decreases the hydrophobic cation current and so forth. So and the recovery is just about the time course we know that the pump can recover, which actually is a trace here in the capacitive signal. You can see how the pumps are recovering there. Okay, so it looks like the membrane is talking to the pumps, and the pumps can talk to the membrane. Now, how could that be? Well, this is a freeze fracture of cardiac sarcolemma. You see all those dots in there? That's at least 10% of the area of the membrane. Those are very probably the pumps. 2,000 roughly per square micron anyway in mice. So one could at least imagine the conformation changes of the pump could actually have influence on the membrane.
Sorry, why are those the pumps and
conceivable.
not just all membrane protein?
They would, but if you just change specifically calcium channels or potassium channels, that would only be a few of those dots. So you wouldn't expect it to be a generalizable effect that the whole membrane would feel. See what I mean?
But you're basing it all on the fact you know
Density.
there's much more of the pump.
Yeah,
Okay.
density.
Okay.
So here there might be a calcium channel in here and
Yeah,
a few.
sure.
That's the okay. So now we have that interaction, and or maybe now what we can show is that many different membrane probes feel what's going on. They feel like, in this case, what did we do? Oh, yeah, this is the effect of a calcium transient again. This is the binding of, we can watch the binding of nitrumic acid. That's a chloride channel blocker. That gives a capacitive signal. We can see that the binding increases after the calcium transient when the capacitance increased a little bit, and so forth. And the next thing is we realize this is very metabolism dependent also, the changes we're seeing with the membrane. So we have seen that a small calcium transients make the membrane apparently more disordered, make things bind better, hydrophobic things bind better. And the converse of that is we know that if we make ATP go down a little bit, or at least ADP go up a little bit, 8 millimolar deoxyglucose in the pump, the pump current goes down like this, down, 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 down. And if you look at the fractional inactivation, it's getting bigger and bigger, and then the pump current's totally gone. No K-ATP current yet. This is metabolic inhibition. It is not enough to activate K-ATP. And here's the hydrophobic cation current. Following it doesn't go down to the same extent. It is certainly going down by 50% or more than 50%. So the membrane is changing not only in response to small calcium transients, it's changing in response to modest metabolic inhibition. There's still 10 millimolar ATP in my paper.
Many years ago, Don, Fran Ashcroft's group showed a complete reciprocal dependence of KATP and sodium pump activity on
Was
whatever
it
that.
she or was it, wasn't it, it was Klaus Bendel.
Before that, she did it with, I forget who it was.
Klaus
but
did it
but,
first. I think Klaus, but anyway.
in the
so
80s.
We're very interested.
Anyway, the
of
point
course.
was, but that was sodium pump activity presumably being inhibited by loss of phosphorylation potential. Is that what's going on here?
I
No.
don't think there's any.
You think this is
phosphorylation.
inactivation, not loss of
Yeah, no.
not loss of phosphorylation potential, i.e. the ATP driving
The
of the pump.
ATP driving something.
It's not
it's
product
not-
inhibition of the pump activity.
For sure, in the excise patch, the pump works fine with 0.2 millimolar ATP and 5 millimolar ADP. So we do not believe that this is depletion of ATP at all. However, decreasing ATP and increasing ADP is changing something about the membrane, which affects the pump, and we think also some ion channels. What the heck could be so tightly coupled to ATP and ADP? What in the world could it be? We've been through a bunch of things. We're very interested in the end in something that has been around for 20 years. Happy or not about this, we're very interested in the idea that it's long-chain acyl-CoA in the cell, which is very, very important to regulate lots of things. People have suggested that long-chain acyl-CoA could regulate sodium pumps for a long time. It's not great biochemical work, but looks interesting. And of course, here for the sodium pump, actually it turned out that unsaturated fatty acids were identified by multiple groups to be the holy grail. That, you know, people thought there were endogenous pump inhibitors for a long time, some fancy compound, some fancy steroid, and what several groups came up with was unsaturated fatty acids. So we think it is possible that fatty acids modify the pump and are generated in response to calcium, could inhibit, and we think they can be turned over into acyl-CoA, which may activate. And, as you all know, acyl-CoA does modify the function of many potassium channels that are lipid dependent, also sodium-calcium exchangers. And it depends on the channel exactly what they do. Okay? And be inhibitory or stimulatory. So, just to reiterate what I said, fatty acids react with basically are used by an enzyme to make acyl-CoA. Of course, that's ATP dependent. And then that can be used further to palmitilate things. That's our subsequent interest. So there's three things: fatty acid, acyl-CoA, and palmitilation we're interested in. And does it have anything to do with what we're seeing? Well, never mind that. Where is it? Hey, there's a slide. Who's going next?
There it is.
No. Data. All right, last slide, I promise. Okay, so the bottom line is, and then over. Okay. Okay. If you inhibit, and there are very selective ways, pretty darn good toxins and drugs to inhibit acyl-CoA synthetase. Here's pump. In the presence of a CoA synthetase inhibitor, triacin C at a very reasonable low concentration. That's exchanger, and afterward the pump only gets smaller and smaller and goes away eventually. Exchange current also goes away. And may I only say, okay, this is very powerful stuff, and I will only add to that all these polyphenolic compounds I mentioned, which block the ATP effect, which we found, genistein, quercetin, methylene blue, they all block very powerfully as anything else they've ever been shown to do. They block the long-chain acyl-CoA synthase. So, my time is up. So, let me just say, we're very interested in this system here, that fatty acids, especially unsaturated fatty acids, can be very important messengers. We are thinking that calcium is generating more fatty acids and other messengers that Dr. Gross and I have been talking about for a long, long time, and that we still haven't entirely worked out very important places where they are regulating membrane physiology at ion channels and transporters. And you know, I could show a little more data why we're getting quite sure that the long-chain acyl-CoA synthetase is critical in the effects of physiological calcium transfers. Okay, but what's much more important is to talk to this guy about how calcium can be activating that stuff, because the literature would not be at this point very clear about it. So, most of the work was done by this guy, Peng-Min Liu, who's a fellow in the lab, and by me. And these folks are very wonderful, but mostly working on other things. So that's the bottom line. We think this acyl-CoA signaling is much, much more important in regulating membrane transport still than, it's been suggested 20, 30 years. But I think it's still much more important. And could lysolipids also be playing a role? Yes, but first and foremost, we need to see biochemically what's happening basically with physiological processes.
So I have a question. So both acyl-CoA and ATP can activate an enzyme IPLA2 beta.
Yes, sir.
Okay, which then can change, right, the membrane structure and mechanisms. All right, is there a way to discriminate?
IPLA2 beta,
right, so we
these
have a
mice,
knockout
which
of that, right?
We have in our mind. We have these mice in our lab. But
No,
not
that's
yet.
you have gamma.
Oh, the beta would be
So the beta,
cytoplasmic.
right, so the beta was, right, so the beta binds ATP. So if you leave like myocardial cytosol just by itself, the enzyme activity goes away in about 2 minutes. But if you put in ATP, it's stable on top of a lab bench for 3 weeks. So the enzyme is stabilized and dimerized by ATP. And acyl-CoA will acylate cysteine 651 and activate the enzyme, and activate IPLA2 beta activity.
Well,
So I think you need some IPLA2 knockout mice.
Yeah. I mean, is it, where does all the arachidonic and so forth come from in the heart when you make some calcium transfers? I mean, you think this is it? I guess you think.
So the two places, the two subcellular compartments that are notorious for having arachidonic, one is the mitochondrial membrane, right, and the other is the plasma membrane, right, which has pretty close to 40 or 50% arachidonic acid at the acidic position in the plasma membrane.
But is it? I mean, CPLA2 is also known in the heart. I mean,
Right.
is it really? I mean,
So
we need to say
CPLA2 is much harder to find in the heart. It's very
Right.
prominent in endothelial cells, with muscle cells.
So we need to definitely brainstorm.
So you need tissue-specific knockouts too.
But the effects are so big. And then, what I would really like to be able to do is, of course, duplicate all this stuff in the excise patch. So one would like to see that some fatty acids really inhibit everything that we think is inhibited, see the membrane change the way we think, and then exchange it for acyl-CoAs, maybe arachidonyl acyl-CoA, and see that we can duplicate all this stuff. So these are ideas that Dr. Gross and I were talking about extensively.
1990s.
Yeah. So I spent the whole summer working on the excise patch on this stuff, and we didn't get to a conclusion. But now, when we went back to the pump in the intact myocyte, it's just bizarre that we came back to exactly the same. 20 years, almost. Close. Yeah, probably it is. Excuse
More
me.
than 20.
Oh. Yes, it is.
But are there more ways that you can directly modulate it? Can you just
Right.
put free fatty acids in the pipette?
Needless to say, that is why we want to, of course, we want to try to make everything work in an excise patch, clean. So 3 things are changed in the excise patch, right? The membrane is totally stretched, the cytoskeleton is totally disrupted, and all of the soluble proteins are gone. So that's it. So we will see if we make progress like that. So we're looking for ways. I think we will be very happy if we can do this like in an eye cell myocyte, where then in the eye cell myocyte you actually can really get proteins in through the patch pipette. In a real myocyte, the proteins just don't move. It's just the molecular crowding is just incredible. But eye cell myocytes are virtually like a fibroblast, and then we can do much, much more to manipulate. So, yeah, that would be the next step. We need some much more definitive ways. So, the things that you can get into the myocyte which manipulate this, like cyclodextrins get in very well, and cyclodextrins, beside binding cholesterol, they bind fatty acids and acyl-CoA very well, even at low concentration. That's been a little, positive and helpful, but, not very significant. So the reason we didn't progress 20 years ago, we just decided this is too damn messy to work with. But it didn't go.
Are sodium potassium pumps distributed uniformly in cardiac myocytes, or there is higher concentration in the T-tubules? How does it work?
Right. For sure, they're in T-tubes and on the outer surface. There is claims that, so we have isoforms, of course, 1 and mostly 3, and I get a little 2 maybe. And there are definite claims that certain one of the isoforms, I think 3, I think, is more in the T-tube. I always forget because we're
The
not.
proximity to calcium from everything
Yeah.
is an important factor.
Right.
So
So.
the density is important in the effect on the membrane. So
Yeah.
if it's all localized in the T-tubules, you would expect a system effect.
So,
Can you-
not a lot.
The same sort of relative idea, can you eliminate the responses completely with BAFTA rather than EGTA, so that chelation of...
Yeah, so the EGTA, we are using 0.5 millimolar
Right.
EGTA. Even 5 millimolar EGTA is plenty.
Okay, it will eliminate the response.
Yeah. Right.
Any other comments or questions? All right, thank you very much again.
Yeah.
Been working on things since 1982 and
Nothing happened.
Well, we dropped the ball.